Hello and welcome! This is our first video in our lecture series for the brand new chapter, Chapter 3, Cell Division. I am Miss Delia, and if you're ready, let's get started. So before we go into the content, have a look at the image behind me. So what we have here are some plant cells, as you can tell by their shape, and also the presence of their cell walls. Now, you notice they're not behaving as you normally draw them. They're undergoing cell division, or more specifically, mitosis, where it's dividing to form new cells. In this chapter, we'll focus on cell division, how to describe each stages of cell division, and also their importance. Okay, so first things first, let's establish the concept. Cell division. Cell division is a process where a cell will divide to form new cells. Cell division involves the distribution of identical genetic material or DNA to two daughter cells. Jadi DNA yang sama berasal daripada sel asal, undergo cell division, dua daughter cell akan dapat genetic information atau DNA yang sama. And when we look at eukaryotes like you and me, it will involve karyokinesis, also known as nuclear division, where they divide and distribute the genetic material or DNA. And second, it will involve cytokinesis, which is the division of the cytoplasm. There are two types of cell division in living organisms. The first is mitosis. This occurs in somatic cells. If you don't already know, somatic cells are cells in your body outside of ovum and sperm. So sel-sel kamu selain daripada ovum dengan sperm adalah somatic cell. And the number of chromosomes in each daughter cell will be equal to the number of chromosomes of the parent cell. So sebelum mitosis, bilangan kromosom dia 46. Selepas mitosis, bilangan kromosom dia pun 46. And the second type of cell division is meiosis. This will occur in germ cells. Germ cells is what we refer to your sperm and ovum as. Okay, and the number of chromosomes in each daughter cell after meiosis will be half the number of chromosomes of the parent cell. So kalau sebelum ovum kita terhasil, dia 46. Selepas ovum itu terhasil, setiap sel ovum itu adalah 23. Ya, 23. Okay, so let us move on. All right, let's talk about why it's important to have cell division. Why is it important to have mitosis and meiosis? First of all, in unicellular organisms like amoeba, it is how they asexually reproduce. Pembiakan tanpa seks untuk dalam organism-organism yang unicellular. And cell division is also important for the growth and development of a young multicellular organism. For you, from baby to become small child, to become teenager, to become the adult you are now, your body had to do a lot of mitosis in order to create enough cells, enough tissue for you to develop. And third, in a mature multicellular organism, cell division is important for renewal and repair of tissues. Cell division digunakan untuk memperbaharui dan membaiki tisu-tisu yang terjejas, yang terluka, yang terbati. And last but not least, it is for our organisms to pass genetic information from generation to generation. Dalam arti kata lain, untuk meninggalkan zuriat. Waris. Mewaris, meninggalkan zuriat. Something like that. Okay. So these are the learning outcomes for this chapter. We hope that by the end of this chapter, you will be able to answer questions that are relating to all of these points. Okay, so this chapter is divided into three main subtopics: the cell cycle, mitosis, and meiosis. And we will go straight into 3.1, the cell cycle. If you're ready, let's go. Okay, the cell cycle, what is it? Well, you can say the cell cycle is kind of like the life cycle of a cell. Dia lebih kurang macam kitaran hidup sel. Tapi tolong jangan cakap kitaran hidup sel bila orang minta definisi scientific. Macam mana kita mau explain? Well, this is the diagram we usually use to explain. Biasa kita guna diagram ini. And you can see the stages through which a cell passes from one cell division to the next are collectively referred to as the cell cycle. Daripada dia terhasil, hasil daripada mitosis misalnya, sampailah dia sendiri pun melaksanakan mitosis, satu kitaran itu kita panggil sebagai cell cycle. So the proper definition for cell cycle is this. Ini definisi dia ya. An ordered sequence of events in the life of a cell, from its origin in the division of a parent cell until its own division into two cells. The cell cycle will consist of two main phases. Kalau kamu tengok dalam gambar aja, kamu boleh nampak mana satu fasa dia. You have this one, which is called the interphase, and this is the longest phase of the cell cycle, making up 90% of the cell cycle. And it alternates with the M phase. Alternate means dia berselang-seli dengan M phase. M phase is from here to here, also called the mitotic phase, and it takes up about 10% of the cell cycle. When we look at this very long interphase, we can divide it into three phases of its own. The first one is the G1 phase, or also called the first gap phase. The second one is DNA synthesis, or S phase. And the third is the second gap, or the G2 phase. Biasa kalau kamu tulis G1, S dengan G2, orang tahu lah kamu maksudkan mana satu. Okay, so we're going to go into each of these phases in interphase and look at them in detail. Right, so let's look at G1, or the first gap phase. What happens inside the cell during the first gap phase? First of all, you would have the cell's growth and normal metabolism. Di sini kita akan nampak sel itu it will act like a normal cell. Okay, dia tidak buat apa-apa yang special. Macam dia mainkan peranan dia yang biasa, dia hidup seperti biasa. Hidup dan membesar seperti biasa. Apakah yang dimaksudkan dengan seperti biasa? Kita boleh tengok di sini. It's the longest phase, as you can see. Dia adalah fasa yang paling panjang lah dalam interphase. In this G1 phase, the volume of the cytoplasm increase. You will have synthesis of enzymes along with proteins. You will have organelle synthesis. And the cell will build up a large store of energy. So cytoplasm dia memperbanyak. Enzyme terhasil, protein terhasil, organelle terhasil, dan dia mengumpul tenaga dalam bentuk ATP. Let's look at the next phase. The next phase is the S phase, or the synthesis phase. In this phase, it has more special things going on. Sini ada lebih banyak benda yang khas berlaku. First of all, DNA will replicate. DNA akan berganda lah, dia akan salin diri sendiri. Here, histone proteins are synthesized. The cell can make duplicate copies of its chromosomes to form sister chromatid. So, dia daripada satu salinan saja untuk setiap kromosom, dia sekarang boleh ada dua. The centrioles will also appear during this stage. Okay, so let's look at this one. Why are we specifically talking about histone proteins? Well, we're talking about histone proteins because if you remember, your DNA in eukaryotes are associated with histone protein. Ini apa yang saya cakap dalam chapter 1, ada cakap sedikit. Chapter 2, ada cakap sedikit. Okay, alright. So let us go on into the third phase in the cell cycle, which is the G2, or second gap phase. So, in this one, it's a little bit different from G1. Dia tidaklah 100% sama, ada beza sedikit. In the G2 phase, the energy stores of the cells will be increased. It will continue to manufacture proteins and cytoplasmic organelles. It will synthesize microtubules. Okay, so sini dia jana tenaga dalam bentuk ATP. Dia masih hasilkan protein, masih hasilkan organelle. Tapi yang special di sini kamu nampak dia mula menghasilkan microtubule. Dan pada masa yang sama, at the same time, it will also replicate the centrosomes and centrioles. Alright, so what do I need to mention here? Let's talk about the microtubules. Apakah benda yang dimaksudkan dengan microtubule? Nanti bila kita tengok gambar raja ini lagi, kamu akan nampak apa yang kamu mungkin pernah belajar sebagai spindle fiber ataupun, I don't know if I'm saying it right, gentian gelendong, I think, spindle fibers, okay, that come from the centrosome or centriole and attach to the chromosome. So, ada microtubule ataupun spindle fiber, nama yang berbeza untuk merujuk kepada benda yang sama. Dia sama ada melekat pada centromere kromosom ataupun dia melekat kepada benda lain dalam sel. Okay, so if it attaches to the chromosome, we call it the kinetochore microtubule. If not, we call it spindle microtubule atau ada nama lain nanti. What is the centrosome and centriole? Centrosome dengan centriole merujuk kepada ini. Centrosome, centrioles. Okay, I think that is it for second gap phase. And last but not least, we will talk about the M phase. What's the M phase? M phase, the mitotic phase, includes both mitosis and cytokinesis. And it is usually the shortest part of the cell cycle. It's about 10% of the life cycle of the cell. Okay. The mitosis or M phase is usually broken down into 5 stages, which is the prophase, prometaphase, metaphase, anaphase, telophase. Ada buku, ada sumber yang bagi kamu 5 stage, tapi untuk silibus kamu kita fokus kepada 4 stage saja. Jadi dalam silibus matrix ni kita tidak berapa tekankan prometaphase. This mitosis or mitotic phase will overlap with the later stages of mitosis and cytokinesis, which completes the mitokinetic phase. Mitotic phase. So, dia daripada karyokinesis sekali dengan cytokinesis. Okay. So, we talk about there being many stages, many phases in a cell cycle. Tapi macam mana dia tahu? Macam mana sel itu tahu bahawa dia bersedia untuk beralih dari G1 phase kepada S phase? Macam mana dia tahu dia bersedia untuk beralih dari G2 phase kepada M phase? Macam mana dia tahu dia bersedia untuk menghabiskan M phase? The answer is that the cell will have its own checkpoints. So dia ada checkpoint atau cara sel itu sendiri untuk melihat bahawa untuk sel itu melihat sama ada dia bersedia untuk masuk ke fasa yang seterusnya atau tidak. There are 3 main checkpoints. Okay, ada 3 checkpoint yang biasa kita belajar. Memang ada lebih dari 3, tapi dalam silibus kita fokus kepada 3 saja. Those 3 checkpoints are G1 checkpoint. G1, which is right here. Dia lebih kurang belum sampai S phase lagi lah. Macam 3/4 begitu. Usually at this phase, it will check if it has enough ATP, if the cytoplasm is enough, if the organelles are enough. The second checkpoint we will talk about is the G2 checkpoint. G2 checkpoint is somewhere here. I think this one, it checks more or less the same things. Dia tengok sel itu cukup keperluan dia ke tidak. And the third one is the M checkpoint, also sometimes called the spindle checkpoint, which is in the middle of mitosis. So this is to make sure that it is ready to complete mitosis with no problems. We don't go into detail about cell cycle checkpoints in your current syllabus, but you are free to go and learn about it online. So here I have a link to this website. You might have already heard about it, Khan Academy. So go ahead and read your notes if you want to learn more or find out more on YouTube or other online resources. Okay? Feel free to explore on your own time. Let's talk a little bit about more checkpoints. What happens if your checkpoints fail? What happens to your cell or to your whole body? You can get cancer. Okay, kalau checkpoint yang saya cakap tadi, kalau dia tidak berfungsi dengan betul, ataupun kalau sel kamu tidak dengar cakap. Okay, kalau kamu nampak ayat dia di sini, cancer cells do not heed the normal signals that regulate the cell cycle. Heed means dengar cakap. Kalau kita ada sel-sel cancer, that means your cancer cell tidak dengar cakap. Walaupun dia belum bersedia untuk masuk S phase, dia masuk saja, dia lantak saja dia masuk. That regulate the cell cycle. If you have these cancer cells, they can go on dividing indefinitely in culture. Dia boleh terus membiak, terus hidup, sampai bila-bila, if they are given a continual supply of nutrients. Sekiranya dia diberi supply sumber nutrien yang mencukupi. In essence, they can be immortal. Dia boleh hidup selama-lamanya kalau diberi zat nutrien-nutrien yang diperlukan. So we have a very striking example of a cell line that has been reproducing in culture since 1951, which is called HeLa cell. This HeLa cell memang sampai sekarang masih hidup sejak dia dikultur. The original source is from a tumor that was removed from a woman called Henrietta Lacks. So memang ini adalah sel manusia yang hidup di dalam makmal. Wanita tu nama dia Henrietta Lacks. Okay, beliau sudah meninggal lah. Tapi sel dia sekarang masih hidup. Kamu boleh belajar pasal fakta ni dalam website ni. We have this blog. And this is the lady. Inilah beliau. Okay, ini Henrietta Lacks, 1920 to 1951. So she died quite early. And over here, you have the image of her cell still alive in the lab. And they reproduce very easily. So, this one has been modified. Ini dia punya sel sudah modified supaya dia boleh berwarna di bawah mikroskop. This has some ethical issues as well. Okay, ada isu etika dan perikemanusiaan dalam bab HeLa cells ni. So if you go and read about this, or if you watch any videos, read any articles about this one, try to think about what are the ethical issues here and what could have been done better. Okay, so that is extra lah. You don't have to do it, but if you do, that would be fun. Okay, so just a very quick summary. Okay, kita rumuskan secara ringkas. Cell cycle ada interface, ada M phase. Dalam interface ada G1, ada S, ada G2 phase. Mitotic phase termasuklah mitosis dengan cytokinesis. And in the next lessons, we'll go into 3.2 and 3.3. So that is as much as I would like to say for now. So now what should you do? As usual, you should make your own notes. And please open the tutorial questions for chapter 3. In the tutorial questions, go to part B, structured question, and ask the question number 1. Jawab soalan nomor 1, nanti kita akan bincang bila kita masuk dalam kelas tutorial. That's it. All from me. Thank you very much and happy learning. And I will see you guys again soon. Bye!